small housekeeping announcement for all of you. I request all the participants to rename their device with the name they have registered for the workshop. It will help us generate participation certificate for all of you. Thank you so much all for joining. Dr. Shetty, the floor is open for you now. Thank you.
Thank you. Thank you very much, Manisha, for the introduction and kind words. Today, indeed, is the fourth and the penultimate day of the international workshop on translational research, taking novel drugs from lab to clinic concepts and case studies. We embark upon a very important topic wherein the outcome basically results in the make or break of the candidate and even for the company proposing the drug. Basically, it is said that whenever there are actions, there is bound to be opposite or adverse reactions. In discovery research, when actions is all about the proof of concept, proof of principle, and pharmacodynamics, the reaction aspect is all about the safety. And today, we try to cover this particular facet of the drug discovery. It indeed is a pleasure on behalf of Drugs and Neglected Disease Initiative, or DNDi, and its associates to invite you all to this very interesting session entitled Toxicology and Toxicokinetics, wherein we will be discussing extensively on the nuances of toxicity and its assessments. As you all know, toxicity is a science that has evolved over the years through incidences, hence resulting in improvements in safety assessment with focus more on precision in identifying the potential risk using different strategies and tools, including kinetics, markers, etc., to get the right perspective on the mechanisms of action of the undesired effect. For this session, we have very distinguished and esteemed speaker who have made a mark in related subject, and from their repertoire will enlighten on various aspects of safety assessment, which surely will kindle a renewed enthusiasm in the topic. Now, I would like to introduce our first speaker, Dr. Venkata Mukavalli, and his topic of discussion will be Introduction to GLP Toxicology. Dr. Venkata Mukavalli is a drug metabolism and pharmacokinetic executive with pharmaceutical R&D and CRO experience. Venkata has PhD in DMPK, MS in analytical chemistry, and MS in organic chemistry. He has supervised and mentored in vitro DMPK, ADME, in vivo pharmacokinetics, toxicokinetics, bioanalytical mass spectrometry, and business operations groups. He is a central conduit in collaborating with project teams for lead selections optimization across various therapeutic areas. He has authored and reviewed developmental plans and IND documents for first-in-human clinical trial approvals, published innovative findings in 23 peer-reviewed articles, contributed to book chapters and 2 patent applications. So I invite Dr. Rao to make his presentation.
Thank you, Dr. Shetty. I'm now going to share my screen. Are you able to see my slides?
Yes, yes.
And I'm audible?
You are.
Okay. Thank you so much. Thank you so much for the organizers for giving me this opportunity to meet and discuss on a very important and critical topic. Give me a minute. Okay. So I will continue from yesterday's session. Yesterday's session was very informative, and personally, it was very important for me for 3 reasons. One, my favorite reporting manager, Anirudh, was presenting. Then second, my favorite teacher, Amen, was presenting, who doesn't like his students' question, but he loves it. Third, a very well-known and respected toxicologist, Dr. K.S. Rao, was there presenting on safety pharmacology. Personally, I worked with KS, and I also, for me as a person, he's a personal trainer for me, because many times he used to say, Rao, let us walk to the next building. And he used to just walk, but to keep up the speed with him, I had to sprint. Having said that, they have sown the seeds for today's presentation for me. I'll be building on the concepts and the examples that they have given. And the outline of my presentation is I will discuss about various classification systems, what are the early indications for the tox team, about the bioanalytical method validation and how we can kind of combine that with the metabolites in safety testing guidance document, and what are the key toxicokinetic parameters, and how does the CYP inhibition or induction kind of help us understand the TK data. So typically we start with property-based selection, where we are having the structural properties, molecular weight, hydrogen bonding, PSA, PK of the compound. When these structural properties interact with the physical environment, it gives rise to physicochemical properties like solubility, permeability, and chemical stability. When these structural properties interact with proteins, it gives rise to biochemical properties like metabolism, transporter affinity, binding, and target affinity. When these two aspects interact in a living system, it gives rise to ADMET, which is absorption, distribution, metabolism, excretion, and toxicity. So based on the physicochemical properties, a classification system was proposed, which is called as biopharmaceutics classification system. In this classification system, we consider solubility and permeability. For compounds which are in class 1 and 3, those compounds show high solubility, and compounds which are in class 1 and 2, they show high permeability. So what is the take-home message for a drug discovery and development team? So previously it was thought of that all the permeability happens because of passive permeability or a combination of passive and active transport. But based on the latest data and the deorphanization of the transporters, it is believed that all the transport is through some transporters that are involved. So compounds which are in class 1 and 2, they will not have any issues with respect to permeability. But compounds which are in class 3 and 4, even if we increase the dose in toxicokinetic studies, we will not see a dose proportional increase. So it is like kinetically derived maximum dose because of permeability being the rate limiting step. Now to this classification system of solubility and permeability, a biochemical aspect or property was added, which is metabolism. So class 1 and 2 compounds, they are highly metabolized. Class 3 and 4, I think there is some disturbance. I would request the participants to
Yeah.
mute their speaker. Thank you. So class.
Sir, I have muted everyone. I just request you to unmute yourself, please.
Okay.
Dr. Rao, you
Thank
have to
you,
unmute
Jayashree.
yourself. Yeah, okay.
Thank you. So class 3 and 4 compounds, they are eliminated unchanged. So again, what is there for us to understand and learn? So for class 1 and 2 compounds, we will see metabolites, and these metabolites may be pharmacologically active or inactive. And when we consider cases of class 3 and class 4 compounds, either the compound may be covalently binding and producing some toxic effects, or it is producing reactive metabolites which are in so small a quantity that we may miss. And why do we miss it? Because of the bioanalytical acceptance criteria. In bioanalysis, we say anything within plus minus 15%, it is more or less similar. So the range is 85 to 115%. If I'm doing a metabolic stability assay and I say that the compound is 10% degraded, within the bioanalytical variation, it is more or less similar, but it may be producing a metabolite which may be toxic. So this is something that we need to be very, very cognizant. There is one more classification system called as refined dullability classification system that pertains to class 2 compounds, and mostly class 2 compounds are those things that we often see in drug discovery and development. So things like micronization, working with pH, or supersaturation, or precipitating agents is going to help us to ensure that the compound is soluble and gets into the systemic circulation. Then, in addition to metabolism, another biochemical property got added, which is transporters. So now based on transporters, again, the classification system is now like classified into 6 different groups based on the molecular weight, less than 400, greater than 400, whether they are acids, zwitterions, bases, neutrals, and whether they are substrate for hepatic uptake transporters, or they are substrate for renal transporters. So very key and important take-home message from this is, as the science is evolving, we need to understand the enzyme transporter interplay. And when we are talking about transporters, these are transporting compounds which are endogenous compounds. So in addition to studying the parent, metabolites, reactive intermediates, we need to also take care of the biomarkers that are there. So now I'm showing a typical screening cascade that we use in drug discovery and development. We start with Lipinski and Weber rule, which are like in silico properties, or in silico data that guides. Then solubility and permeability, which are physicochemical properties, and then metabolism, transporter, MET-ID, formulation development, and then PK studies. So I'll start with a case study. I was on a project where we were advancing a compound. Solubility of the compound was very high. It was quite good. Permeability was also very good, but the efflux ratio was less than 2, which is also a good property. That means it is not a substrate for any efflux transporter. When we started with metabolic stability studies, the compound was chewed up in 10 minutes in all 4 species: human, dog, rat, and mouse. So this is the point where I went back to the project team. I said, are you sure that in vitro and in vivo you are seeing very good efficacy? And the pharmacological lead confirmed. So I said, I don't want to go through the screening cascade, but I want to do a metabolite identification study. We did a metabolite identification study where we identified the phase 1 and phase 2 metabolite. So we quickly synthesized it, confirmed it, and gave it to the pharmacologist to test for the in vitro and in vivo efficacy. They came back saying that the metabolites are also active. So from then on, the studies, all the studies that we were doing, we had the parent and the 2 metabolites in the method, in the quantitation method. We validated the methods, did the toxicology study, and the compound was successfully out-licensed. The reason why I'm giving this example is, if we would have missed not looking for the metabolites, it would have been that the compound has advanced, we have done toxicology studies, then finally at one point of time, we would have gone back and looked for the metabolites, metabolite synthesis, again going through the same steps. So in a way, we saved at least a years of time by doing the MET-ID upfront and kind of looking for them also. Now, the critical aspect, and what Eamon was referring to yesterday, that PBPK modeling is very close to his art. This is how we do it. We do in vitro studies using liver microsomes in mouse, rat, dog, monkey, and human, and use the data to predict how it is going to pan out in the preclinical species PK, TK. And we have the in vitro data. So in the jigsaw puzzle, we have 3 data sets, and we can use the simulation and modeling to see how it is going to pan out in the human species. So yesterday, there was a question about how do we choose the preclinical and preclinical rodent and non-rodent species for toxicokinetic studies. In addition to whatever case explained, there is one more key consideration, that is the metabolite identification. So we need to choose the rodent and non-rodent in such a way that the metabolites are covered when we go into humans. For example, if I'm seeing a metabolite in mouse and rat, which is also seen in human, but it is not seen in dog, but seen in monkey, then I would prefer to go with monkey as a species for tox studies, because it is going to cover for my metabolite, which I'm going to see in humans. Here, in addition to quantitation of the metabolites, it is very important that we run the samples on high resolution mass spec. When I used to work with patient samples who are very critically ill, I used to run samples on HRMS at that point, we may not know what are the metabolites, what are the biomarkers that we are going to work in the future. So HRMS actually helps as a data bank, and it is very easy for us to go back retrospectively and look for those metabolites or biomarkers, which may be kind of discovered 2 years down the line. So again, from my side, recommendation, run the samples from PK/TK studies on HRMS, have it as a database, and you can go back retrospectively and see what kind of data or biomarkers or metabolites are present in that. Next, I'm moving to method validation. So when we are validating the method, we need to have the parent, also the metabolites and the biomarker. We need to be very cognizant that we encompass all these three aspects when we are doing the method development and validation. Typically, we start with method development. We develop the method, we validate as per the guidance documents, and then we put it to operational use. Sometimes we validate the method at one place and transfer it to another place. The robustness of the method, validated method, comes into play when we put it to use. Many times we have to increase or decrease the injection volume or change the column or make small adjustments, because of which we have to go back and run one accuracy and precision batch. Or if it is not at all working, we have to go back, redevelop the method. So what are the key ingredients when we do the method validation? We need to be very particular about the sensitivity of the method. What is the limit of detection? What is the limit of quantification? It should be selective and specific for those analytes. We have to run 3 accuracy and precision batches, and we have to see what is the range. We need to check the dilution, assess the dilution integrity in toxicology studies, because the doses are very high. We will end up diluting the samples a lot. So we need to ensure that the dilution integrity is there. We have to establish the stability of the samples, and we have to analyze the samples and reanalyze the samples using the same method. Now, it is well defined, and we have very good handle on how we do the method validation, but there are some key considerations that I will bring out here. The first one is which matrix to use, whether it should be blood or plasma. So based on the distribution studies, we have to see if the compound is partitioning into RBCs a lot, and now the latest data shows that RBCs have some transporters and pardon. So RBCs have some transporters. So it is very important that we assess whether the compound is going and sitting in the RBCs, and if blood is going to be a better matrix compared to plasma. The second aspect, which I would say, is mostly we work with many prodrugs or compounds which have ester groups. They form a lot of metabolites, and these compounds are sometimes very unstable in plasma. If we collect the plasma, they may degrade in the plasma. So we need to be very cognizant as to if we need to add any stabilizing agent for the analyte or the metabolites. Personally, I have seen on standing, plasma loses CO2, and because of loss of CO2, the pH of the plasma shifts. We expect it to be 7.4, but when I measured, many times it has been 8.2, 8.5. So it is very important and critical to understand the characteristic pK of the compound, because I have seen that the binding characteristics change. Even the species that is present at pH 7.4 versus 8.2 or 8.5 is different. So it is very important upfront to understand the analytes that we are working with, what is their pK value, how their structure is going to change. For example, lactones, if we are working in the basic environment 7.4 or even higher, they are going to break open and form acids. So most important thing that I have seen is scientists sometimes miss to understand or address these gaps, and whatever PK or TK results that we get are anomalous. And these are some of the key considerations. And when I talk about method validation, one of the key considerations that I see is the LLOQ of the method. If the LLOQ of the method in the front CC calibration curve and the back calibration curve and the 6 QC samples and equal to 6 replicates that we run, if it is working out, that means the method is quite robust, because LMH QCs, that is low, medium, high, are going to work out. Now, once we have established the method, we analyze the samples from toxicokinetics studies, whether it is rat, dog, monkey, or mouse. And typically, the profile that we get is like this. On the y-axis is the concentration, on the x-axis is the time. And the day 1 profile is like this. Day 14, day 28 can exactly mimic the black line. Or day 14 can be higher, or it can be lower. How do we understand this data, and how do we mitigate if there are any issues? So the effective concentration is from the pharmacology that we get from the PD studies. So for the compound to be effective, we need to have concentration which is over this line. So for this particular compound, it is over this line and it is a good exposure. But over time, if you see an induction, then the compound concentration is falling down, which is like therapeutic failure. This is the tox threshold, again, NOAEL, no adverse effect level. And Nigel is going to show a very nice example of this, why this is important, and if this line and this line comes closer, why it is not a very desirable property. And because of inhibition or inhibition of any clearance component, the concentration goes over the toxic concentration, and how to understand and address and mitigate that issue. I'll start with CYP inhibition. Again, Case touched about this example yesterday, the example of terfenadine when it was taken along with grapefruit juice. This is not grape, it is grapefruit, which is a little bigger than the oranges that we see in the market. So when the patients took terfenadine and along with that they drank a glass of grapefruit juice, they had these cardiac events where some people also died because of the QT prolongation. And it was because of a component which is present in grapefruit called bergamotin. It inhibits a cytochrome P450 enzyme, which doesn't allow terfenadine to go into fexofenadine. Here I'm showing the terfenadine concentration versus time, how when taken with water and how when taken with grapefruit juice, the PK parameters differ. Bottom, it is an example where with increase in concentration of terfenadine, there is an increase in QT prolongation, which ultimately leads to heart attack. So this compound was actually withdrawn from the market because of the unfortunate events that were seen in the patient population, but its metabolite is still in the market. So in case we are going to see these kinds of DDIs, can we predict upfront using in vitro studies? The answer is yes. A very simple study, I will walk you through this slide. We take an enzyme, we incubate with the substrate, which is a prototypical substrate. For example, for 3A4, let's take midazolam. It forms a product called 1-hydroxymidazolam. So when we incubate this reaction with our test article, we may see an inhibition, and the inhibition profile is shown in red. So if we continue incubate this test article only with the enzyme without any cofactor, it may inhibit the enzyme, which is again because of the covalent binding of the test article to the enzyme. And if we incubate this test article with enzyme along with the cofactor, it forms metabolites, and those metabolites may show the inhibition that is here. So when we do this CYP inhibition, mechanism-based inhibition, and time-dependent inhibition, we see 3 graphs. Either they may overlap or they may separate out. So what it is telling me is, in case I see a compound which is metabolically stable, but it shows a profile like green, which shows that the compound may bind to the enzyme and may cause irreversible damage to the enzyme. This is again something that we need to be cognizant. Again, for a compound which metabolizes very less, but forms metabolites which show a very, very potent inhibition, then we need to be cognizant. Now an example, did I see this kind of profile and did it ring alarm bells in my mind? Yes, the answer is yes. And I did sense based on these experiments that it is going to be toxic in toxicokinetic studies. And Stephanie is going to deal with an example. I saw similar kind of off-target toxic impact. And now, what is the consequence of that? The consequence of that is the dose that we will give it in toxicokinetic studies are in clinic decreases. I'll walk you through this graph. So very, very important when we are in drug discovery development to understand whether we are working with a reactive chemical or a discriminating drug. PAIN stands for pan-assay inhibitory compounds. So these compounds react with everything, whether it is enzyme, and we may read out and say that this compound is a very good drug candidate, but on the hindsight, it may be very, very toxic, and the toxicity may be causing those effects. So I will introduce to you a rule of two, which is based on the log P of the compound, which is a structural property, and the daily dose that we give. As you can see in this quadrant, most of the pink dots are those compounds which showed a lot of toxicity. Adverse drugs reactions are DILI, that is liver injury in either the animals or in patients. These are 44 which are here, and only 2 compounds which are positive. So if a dose is more than 100 milligram, then the probability of seeing a toxicity is too high, and if the log P is greater than 3. So this pie chart shows those compounds which have been withdrawn from the market because of the toxic events. If it is more than 100 milligram of dose, 84% are withdrawn. Between 10 and 100, around 16%. If it is less than 10, it is 0%. Now the marriage is between the toxicity, the PK, and the PD. So the drugs which are in the market with black box warnings, if the dose is greater than 100 milligram, 81% of the compounds, and between 10 and 100, it is 19% of the compounds. So these graphs again show the correlation between log P and Cmax and daily dose and Cmax. Again, the Cmax is very important. If it is more than 1 micromolar, we can see most of the compounds which are showing the toxicity. So this was about the inhibition and how it can manifest, and using the in vitro assays, how we can understand and mitigate the situation. So the overall summary from here is, if the compound or the metabolites are toxic, it is going to limit the dose that we will give in toxicokinetic studies, and at a very low dose we will see toxic effects. Next, I will move on to CYP induction study, where we see a therapeutic failure. Digoxin was given with St. John's wort, which is an antidepressant and anti-inflammatory. And over a period of time, the concentration of digoxin decreased. So St. John's wort, which induces many of the CYPs and transporters, may impact or interact with 80% of the compounds. When I talk about CYP induction, 10 years back, it was only CYPs that it will perturb. For example, AHR, CAR, and PXR are nuclear receptors which are available in the body, and they are responsible for the induction of 1A2, 2B6, and 3A4. These are the cytochrome P450s which are responsible for the metabolism. But the recent studies shows that, again, this picture is from notes of Eamon's class. As I mentioned, he's a teacher also. So in addition to CYPs, it may also perturb the transporters. For some transporters, it may increase. For some transporters, the concentration may decrease once we treat it with a test article. So
Dr.
to
Rao,
summarize,
Nitin Shetty here. You got 5 minutes to complete your presentation.
Yes. So to summarize, it is just not the phase 1 enzymes or metabolism enzymes. It is also the transporters that get perturbed when we treat it with test article. Here I'm showing the species and organ expression of different CYPs between the human, mouse, rat, dog, and monkey. The point, the key highlight here is when we are translating the data that we generate in the rodent and non-rodent species, how it is going to pan out in humans because of differences in CYPs and transporters in various organs. As I mentioned, transporters and CYPs are not only there to take care of the exogenous compounds, but also for endogenous compounds. And for these transporters, I will take example of transporters. For example, renal transporters, OCT2, MATE1, MATE2K, there is a creatinine, which is a very good surrogate. And Ayman mentioned about translatable biomarkers. So these biomarkers can be used in preclinical species. In addition to the analytes, we can look for these biomarkers also, and the same can be used in clinical studies to understand what kind of impact it is showing, whether it is for bile acids or something like creatinine. Moving further, the gender differences also come into play when we are trying to interpret the toxicokinetic data. These are the trend CYPs and transporters that show the maximum gender difference between males and females. So these are some endogenous compounds or compounds which we dose, and the ones in red are the diseases. So the key point I want to make is 8 out of the 10 drugs that were withdrawn from the market showed adverse drug events more in women compared to men. So that's why when we do toxicology studies, we take male and female gender and then do the toxicity studies. So to summarize, when we do toxicokinetic studies and we do the method development and validation, we need to be very cognizant about the analyte, metabolites, whether reactive, non-reactive, pharmacologically active, not active. In addition to that, we need to also be cognizant of the biomarker that we are using. If there is a disease state, then what kind of biomarkers that we can select. Based on the inhibition profile that we see in toxicokinetic studies, whether it is decreasing the activity of enzymes and transporters, what is the impact on drug concentration, whether it is inducing, increasing or decreasing the activity of enzyme transporter, and what is the how it is impacting the drug concentration. In addition to that, what is this impact of species difference, gender difference, organ difference, and the polymorphisms that we see. With that, I will hand over back to Nitin, and if there are any questions we can take.
Thank you, Dr. Rao. I think there are two questions that has come up. The first one is, if accumulation properties of compounds seen in both rodent and non-rodent, however, in phase 0 studies, there is no such trend seen, then how do we convince regulators the feasibility of the human dose?
Very good question. So if there is no, as I mentioned, the current trend is to go for PBPK modeling, and PBPK modeling is coming to our rescue, I would say, in a very big way, whether it is SimCip or GastroPlus. So as I mentioned, we do in vitro studies. We take the in vitro data and see how it is going to pan out in PK. We do the actual PK studies and then correlate whether the PK in silico and the actual PK is correlating. The same thing is done for human. We use the in vitro data and see what kind of human data we anticipate. Armed with this data, we go to regulators and we present what is going to be those. Below, I have shown some allometric scaling based on the mouse, rat, and dog data. This is what was predicted for human based on the clearance and volume of distribution. Once we know the clearance is dosed by AUC. So once we understand this is the AUC that we want in the clinic to actually be over the efficacious concentration. So once we know the exposure and the Cmax, Tmax that we want, we go armed with this data to the regulators to convince them.
Thank you. One more question. How will you differentiate between a PGP substrate and a PGP inhibitor?
Again, very good question. This is something that I often see that we get wrong. The first way of doing it is to do the permeability study, which I have shown here. when we do a KECO2 permeability study, we get an efflux ratio. If the efflux ratio is greater than 2, that means it is a substrate of one of the efflux transporter that is present in KECO2. It can be PGP, BCRP, MRP2. The other way of doing it, this is about the inhibition. If in the KECO2 study, we get an efflux ratio of less than 0.5, that means it is a substrate of some of the uptake transporter that is present in the KECO2. So typically when we do transporter studies, I recommend starting with inhibition study. We have 9 transporters that are recommended by FDA, 11 by EMA. Once we do the inhibition study, and if the compound scores as an inhibitor, it is going to be a substrate for that transporter. And now there is always a danger of the compound being a substrate at lower concentration and being an inhibitor at higher concentration, or vice versa. So it is very important to do both the substrate and inhibition studies and see at what concentrations they are acting as substrate and inhibition, and then take an informed decision.
Thank you. And one more last question. I think we can pick up on this question. For MABs, do you see any toxicokinetic studies on how it is to be done and interpreted, especially the in vitro part?
Again, as KS mentioned yesterday, this field is evolving, and as the data is evolving, we are working towards understanding the compound. And because the doses are less, again, Anirudh also mentioned it is TMDD, target mediated drug discovery, that we are kind of seeing. So again, it depends on the large molecule that we are working on, and based on the data and case-to-case basis, we need to see. If you ask me if these CYP inhibition and transporter studies are applicable, yes, for large molecules, I do recommend, and even the companies, pharma companies, are doing the transporter inhibition and CYP inhibition studies, because again, it is very difficult to understand the substrate modality. But at least in these inhibition modalities, we can understand, because we are not measuring the antibody, but we are measuring the prototrichial substrates, metabolites, or inhibitors that we are using. So yes, we do CYP inhibition and transporter inhibition studies for large molecules also.
Thank you. Thank you very much, Dr. Rao. I think
thank you, Dr. Shetty. And I would like to thank DNDi team, you, and my boss at Absorption Systems, Dr. Sid Bhupati, for making this happen. Thank you so much.
It was a pleasure having this presentation. So we go on to the next presentation, Introduction to GLP Toxicology by Dr. Senthil Kumar Mariyappan. He is a veterinarian by training with masters in veterinary pathology with 15 years experience in pharmaceutical industry. He is an expert leading development of new molecules, differentiating medicines, standards, and complex generics. During the early part of his career, he has worked in CROs in India as a toxicology study director. Later, he established himself as a project toxicologist in development of NCs and generics in India before relocating to Germany in 2016. He is currently working for Bayer Pharmaceuticals in Germany, where he is associated with supporting development projects of diverse modalities, small and large molecules, as toxicology project lead. Dr. Senthil Kumar, I request you to initiate your presentation.
Thank you, Dr. Shetty, for the nice introduction. I welcome you all for this part of the 5-day international workshop, where I am here to present this topic, Introduction to GLP Toxicology. And I'm glad that this topic finds a place on this 4th day of this 5-day webinar series, where you might have already listened to various topics regarding how to take a novel drug from lab to clinic. In the previous days, you might have listened to various sessions, right from lead optimization to pharmaceutical development, and then the PK and PD relationship, and the modeling approaches which is available behind PK and PD relationship. And this is the right time now to listen to some basic concepts of toxicology testing, with a particular focus on what kind of innovative approaches can be adopted in the routine toxicology testing paradigm. With that prelude, I will go into the presentation. Just give me a moment, start moving. Okay. So in the next 20 to 25 minutes, I will be walking you through some of the topics as listed down here. To start with, from the different phases of drug development and the flow of a small molecule drug discovery and development project from a toxicology perspective, with a particular scope on what kind of innovative testing approaches can be adopted, both during the early safety assessment part as well as during the regulatory toxicology part, which is nothing but the period when we do IND enabling toxicology, GLP toxicology studies. And then we'll touch a little bit on the key aspects of in vivo toxicity testing, where there are a lot of scope lies in terms of improving the way we do such kind of studies to come out with the best translational outcome before a molecule is handed over to the clinics. And then I'll touch a little bit on the role of toxicologists in enabling first-in-man study before I summarize my talk, followed by Q&A session. So looking into the phases of drug discovery and development, I can broadly categorize into 3 different stages. The first one is preclinical research stage, and then followed by clinical research stage. And once the molecule is approved, it goes into the market authorization and market access phase, which is nothing but the launch and access. In preclinical research stage, you'll have 2 different phases. One is the basic research and followed by a discovery research phase. And when the molecule goes into the clinics, it has 3 different phases of testing. And then once it's approved, it goes into the post-marketing, which is phase 4 of the drug cycle. And the focus of our presentation today and also on this 5-day webinar series is taking a molecule from the basic research phase till handing it over to the clinics. And that's where our focus of presentation is going to be, where the toxicological testing requirements, which will be typically needed during the basic research phase as well as the discovery research stage. Toxicological testing follows typically a tiered screening approach right from the target selection stage till the first-in-man human clinical trial. At the target selection stage, it's a responsibility of the toxicologist to do a detailed target safety assessment. I will come to this part a little later in one of the upcoming slides. And during the lead compound identification and optimization stage, we will be typically required to do some kind of in vitro early safety assessment studies, which include, but not limited to, cytotoxicity assays, mitotoxicity assays, and off-target profiling, HERG, which is a part of safety pharmacology, and gene tox profiling. Here, this means screening gene tox and not a full-blown GLP gene tox program, and then some kind of mechanistic organ toxicology. And after completing this in vitro toxicology stage, when the molecule proceeds into the in vivo exploratory safety assessment phase, we will be expected to do some kind of exploratory toxicology, including some omics technology and mechanistic toxicology as well. Combined together, these 2 phases will contribute the early safety assessment of toxicological testing. And once the molecule moves into the regulatory toxicology stage, we will be expected to do these typical first-in-human enabling non-clinical safety studies. And this, after completing this, an IND or investigational new drug application is filed to seek approval for conducting the phase 1 trial in humans. Okay, and remember that throughout all the phases of drug development, toxicologists would have to have effective collaboration with all the key stakeholders in the drug development scenario, and I've highlighted here. And a standard project flow from a toxicology and safety pharmacology with a particular focus on new chemical entity goes something like this. And I've highlighted here the list of studies which would be required at each and every stage, right from discovery, where the lead generation and lead optimization happens, and during the development phase, which is the regulatory testing or toxicology testing phase. And once the molecule is approved here, it goes into the clinical phase, and even then we need to do some kind of studies to make the molecule ready for an NDA application. NDA is nothing but the new drug application. I will not read the kind of studies required here because these slides are available anyhow to you, and also that you can refer the list of studies from any literature. But with the note that the extent and nature of studies listed down here is just for an example, and it will always vary based on the therapeutic modality or the route of administration which you are working upon. But I want to emphasize the role of toxicology does not actually starts from here. It even lies before the drug discovery phase, where toxicologist has to be an integral part of the project team meetings from the early research phase on, where similar to a pharmacologist, a toxicologist should know the target or the disease and the target biology and the chemotype which we are working upon, the indication which we work upon, and what kind of target and the downstream signaling pathway which we are intended to modulate. So all these informations are very, very crucial for a toxicologist to know, because it helps a lot in finding or categorizing an effect which we will see during our typical toxicological studies, to categorize it as either adverse or non-adverse finding. Because it's not unusual that during toxicity studies we tend to see a lot of findings which are just an exaggerated pharmacological response and not really have to be declared as adverse. And this will have a major impact on your NOAEL consideration, which is nothing but the no observed adverse effect level consideration. And that's why it's very crucial for us to know the entire background of the project which we work upon. So coming to why innovation is needed in toxicological testing methods. A novel testing strategy is the need of the hour now, because if you look into literature to see the translatable success of preclinical to clinical phase, you will get mixed response in the literature. Some literature would say that the preclinical findings are not really translatable to clinics, and some say that yes, it is to some extent. And definitely there lies some inaccuracy in terms of extrapolating the findings from preclinical studies to clinics. And that's where a lot need to be done to improvise the way we do the toxicological testing. And this is also with the note that we need to de-risk the safety liabilities which we see in our molecule at every stage of testing. And that's where the need of adopting some innovative non-clinical study designs are needed, which could be more of science-driven approach than just following what is mentioned in guideline. And this gives us enormous confidence when we go into the clinical trials. And there are certain areas of improvement lies in early and regulatory toxicology phase, and some of them are listed down here, and this is going to be the essence of my presentation going forward. So before I go into, before I go into the innovation in regulatory toxicology, I would like to touch upon. I hear some noise. Can someone go on mute?
Please mute yourself, please.
So before I go into the innovation in regulatory toxicology, I would like to briefly touch upon the innovation in early safety assessment phase, because this is one of the key area where we do a lot of in vitro and early in vivo toxicological studies to increase the chance of success when we move into the regulatory toxicology phase. And during this, there are a lot of advanced methodologies or platforms or tools are available, which are typically come under the broad umbrella of what we call as predictive toxicology. And some of them are listed down here. I typically want to put it under 3 different buckets, namely quantitative systems toxicology, then evidence-based toxicology, and then something called new approach methodologies, which are typically here to meant to supplement the existing standard testing strategy. I will not go into the details of it because the content of this particular slide alone will warrant for nearly 1 hour discussion, and that's not going to be the focus of our talk today. So coming to the innovation in regulatory toxicology, I would like to categorize into 2 different approach. One is the evolutionary approach, which is what is currently going on. So there is definitely a need for optimizing the current testing strategy. for which a toxicologist should have a better understanding of the biological and chemical space upon which we are working on. And also there is a need to validate the individual assays which are currently available. Here, animal study was and is the current gold standard upon which all these studies are designed. And then the next approach will be the revolutionary approach, where the aim is to address relevant human physiology in appropriate test system, which means as against using animals, here we tend to use human cells in a 2D or 3D cell culture, or in organ on chip model, or on body on chip model. So these are all some advanced tools which has revolutionized the field of toxicological testing to a greater extent. And here the major emphasis is on human biology. As you can imagine, this is coming up very nicely, but when it comes to the matter of using such approach in regulatory toxicology testing phase, upon which the regulators normally tend to give the approval to go ahead with a clinical study, this is still to see the light of the day, I would say. So at the moment, as we are talking now, animal studies still happen to be the gold standard for toxicity evaluation in regulatory toxicology. And that's why there is a need for us to modify the way we do the current testing paradigm, which means we need to customize the package of first-in-man enabling toxicological studies, which should suit the intended clinical trial. And we should take care that such an approach should meet scientific, regulatory, and operational requirements. Having said that, it is really very important for us to follow the guidance-based recommendations, but we can always supplement these recommendations by appropriate additional measurements or evaluations, which goes totally based on your science-driven thinking. So having laid that background, I would like to concentrate on the key aspects of in vivo toxicity testing, where there is a huge scope of improvement is still there. I will go through one by one in detail in the coming slides. The first and foremost, the aspect of toxicity testing is the species selection. And it's a topic which was asked yesterday to Dr. Rao, and in my previous session also, Dr. Venkata also highlighted to some extent. And I would like to highlight again here that the species justification is a crucial part of toxicological testing. Gone are those days when you think that, okay, a small molecule need to be tested in toxicity program. So I will select rat and dog as the two species of choice because they give, there are a lot of historical control data available, and they are very easy to handle, easily available across CROs. And the test item requirement, if I go for dog, is comparatively lesser when we go for mini pigs or other large species. No, that's not the approach we should have. We should really look into the aspects like the similarities or differences across different preclinical species available when we compare with humans. And this can start from the target characterization part itself. For example, if your target is a receptor, you should look into its expression pattern across different species, its homology, its distribution, and subtypes. And you should also know the physiology and pharmacological comparison across different preclinical species as against the humans. And the most important part is the metabolism and PK. So for example, the metabolite profile, this is what Dr. Venkata pointed out in his lecture previously, that you should compare the in vitro metabolite or the in vivo metabolite profile between different animal species against humans. And then this will really form a basis along with your target characterization to choose the best animal species of choice for your toxicity testing. For small molecules, guideline says that at least one species which is used for toxicity testing, which can either be a rodent or non-rodent, should be pharmacologically relevant. What is meant by pharmacologically relevant? You should see that their target should be expressed in this particular species, and it should also be modulated, meaning in your pharmacology experiments, you should have seen that some experiments should have been done where the target is engaged and modulated to some extent. And that forms the basis for you to select the species. And this is where, again, I would like to reiterate the importance of toxicologists to be in collaboration with scientists like pharmacologists or the DMPK colleagues, and in choosing the best species of choice. And also to highlight that the human risk assessment is always primarily based on the results which we obtain from the most sensitive species for human relevant effects. And something into the dose selection part. Guidelines says that the high dose selection for the pivotal first-in-man enabling studies should be fulfilled based on maximum tolerated dose or maximum feasible dose or the limit dose. A limit dose is something where you can go up to the maximum level. You cannot go more than that, even if that dose is not the maximum tolerated dose. And similarly, a maximum feasible dose is the dose which is practically possible due to some technical reasons. Even if it is not the maximum tolerated dose, if you are not able to go beyond that dose, you have to stop there. And a basic tenet of choosing the dose levels in a toxicity study is that your high dose level should at least address 50 times of the intended human exposure, and the low dose V in Bayer follows the principle that it should represent a low multiple at least to 2 to 3 times of the targeted systemic exposure in humans, and the intermediate dose should be some geometric mean between the low and high dose. So here again we have the PK-PD correlation comes into place, where based on the PK and PD modeling, you will often get to know the predicted human efficacious exposure based on which you can calculate the dose which you need to go into the preliminary dose range finding study in your toxicity program. And also we have to understand one fact here, that as per the basic tenet of toxicology, the high dose should show toxicity, but it's not always true that you see that effect happening. And that's why you always have the liberty to increase your dose level in the subsequent study, taking care not to compromise on the integrity of the study, meaning there should not be that there are more mortalities happening in your high dose in the subsequent studies. And something into the importance of adding additional endpoints and biomarkers. So if you look into the guidelines, you'll get to see that there are certain standard parameters like in-life phase observations, and also the clinical pathology parameters, anatomic and histopathology parameters listed down in guidelines, but that they are just for you to guide through, and that's not the end of it. Based on your compound nature and the indication which you are working upon, you always have the liberty to add additional endpoints or biomarkers, and some of them are listed down here. I will not read through all of them. Just to highlight an example, for if needed, you can also include non-standard microscopic evaluations such as some special stains. For example, if your target organ, if you expect that your target organ is going to be bone, then you can include some additional markers in plasma or serum, for example, cathepsin or some osteocalcin, for example, and also some stains like Vancosa as against the standard HND staining in histopathology can be included, where you can see some useful information coming out of your toxicity study, which would have a direct implication when you go into the clinical trials. And also, if you in your early study, if you observe some organ-related toxicity, you can go for a scientific assessment of the pathophysiology, or by allowing sufficient dose-free period, which is typically known as the recovery or reversibility period. You can have a broader understanding on the organ-related toxicity, which will have an implication on the clinical trial as well. And something into the duration of studies. The duration of toxicological studies depends typically on the anticipated PK/PD characteristics. For example, the half-life of the drug, its nature of binding to the target, or the reversible or irreversible, and also on the persistent pharmacodynamic effects, as well as the specifics of the intended development plan. According to ICH M3, a 2-week study are the minimum duration, but for you to go for an IND filing, you should have definitely done a 4 weeks toxicity study. Why a 4-week study is because based on a meta-analysis, we found that a 4 weeks toxicity study predicts target organs in toxicology study to a greater extent than a standard 2-week study. And that is expressed in the graph on the right side, where you see that in the y-axis, a number of human toxicities are mentioned, And I would like to draw your attention on the third and fourth bar here in green color and blue color. The third one is for the 2-week study, and the fourth one is for the 4 weeks or the 1 month study, where you see that the number of cases upon which the human toxicities are detected almost doubled. But you see that afterwards, there is a stagnant phase here. Where you see when you conduct 3 months or 6 month study, there is a little increase in the case where you can see the human toxicities. But we still have to do these 3 months and 6 month studies because on one end, you still see some toxicities coming up and you do not want to see your compound lying in one of them. And also on the other requirement is that it's still a regulatory requirement you have to do to meet out the clinical duration of treatment. And when you take the duration of treatment into consideration, you should also see that there should be an evidence of biomarker activity happening, which could be in the form of target engagement or target modulation in your preclinical species of choice. But we should also take into consideration that this is not always possible because we do studies in healthy animals and not in diseased models. Sometimes when for some compounds where you are working on vasopressor, for example, you tend to see such hypertension as a target engagement modulation activity happening in healthy animals too. But for other targets, you won't typically tend to see this. And then some thoughts into the route of administration. Route of administration in preclinical studies should always be the same as the intended clinical route of administration. But there are certain situations which might warrant an alternate route in preclinical study. For example, if your compound is poorly soluble or poor absorption is noticed from the vehicle which you use in the toxicology study, you can go for an alternate route. And if the rate of clearance is much higher in toxicology species than which is anticipated in humans, then you can go over an alternate route of administration where you can take care of this difference in rate of clearance. And then finally on the practical feasibility grounds, meaning, for example, if you're developing a transdermal patch and you want to do a 6-month rat study, which will be typically difficult in a rat, so you can then adopt or go for intravenous or subcutaneous studies, taking care the systemic exposure is addressed there. And it's not a surprise that even during the clinical stage of testing, you might see a condition where the route of administration gets changed and you will be then approached to do some kind of bridging non-clinical studies. One such example, which I have witnessed in my career, is that there was a molecule which was primarily intended for intravenous route of administration. All the data was generated for intravenous route of administration, but during clinical stage, there was some issue due to which systemic exposure or there was systemic exposure related toxicity observed, and that's why the route of administration has to be changed to inhalation route of administration, and it is going on well now. And this is one of my favorite slides where I want to touch upon the learnings which we acquire from a drug attrition. And I have given here the the reference from which this information is taken. Basically, 1/3 of the project stopped before progressing to humans during non-clinical testing. And there was one recent publication from 2015. This is a cross-industry collaboration-based publication where some 4 major companies collaborated together, where they analyzed their the development programs, and they concluded that in 40% of the cases where they had to terminate the program, they attribute the reason to non-clinical safety. But this we have to see as a positive measure. This is actually a de-risking measure in the early safety assessment, so that we don't waste our resources and money into some compounds which will not go further into the regulatory toxicology and clinical phase of testing. This is nicely explained in the figures on the right side. On the top panel, you see that number of compounds is listed down in the y-axis, and you see that during the candidate nomination phase, there are 356 compounds which had to be terminated. And then in phase 1, it is 157 compounds, and in phase 2, it is 89 compounds. And if you take the blue portion of this graph alone and elaborate further into in the bottom panel, you see that in the candidate nomination phase, out of 356 compounds, 211 compounds had to be terminated, citing non-clinical toxicology reasons. But again, this is, I would highlight the de-risking measure and not as a failure. And when the molecule goes into phase 1 and phase 2, The reason of termination due to non-clinical toxicology finds the fourth place in both the phases, where the first reason is still being the clinical safety in phase 1 and the clinical efficacy in phase 2. And there are further evidence or further literature available on this aspect. But as a whole, I want to highlight that there is definitely a need to improve the earlier screening paradigms with the goal of earlier de-risking strategy even before we reach the GLP toxicity testing phase, so that we fail early to avoid unnecessary resource and money waste.
Dr. Senthil Kumar, you'll have to finish your presentation in 5 minutes now.
Okay, I'll do that. Thank you, Dr. Shetty. So now, having conducted the toxicology study, we are at a phase of transitioning from non-clinical to clinical. So we need, based on our toxicological studies, we need to identify the hazard or risk, which means identifying and characterizing the target organs in the preclinical species, and then evaluating its relevance for humans. And when the relevance is established, you need to always look into the margins of exposure between toxic effects and the predicted efficacious exposure, and also need to figure out whether such effects are monitorable in the clinics or not. And here, the toxicologists, I would say, have a major role along with the clinicians in designing the clinical protocol, which includes designing or deciding on the subject inclusion and exclusion criteria, stopping criteria, and availability of biomarkers, etc. And yeah, this slide is again highlighting to the points from the previous ones. So I would skip this for now. And yeah, this I would like to again highlight here the importance of a toxicologist along with the DMPK colleagues and clinical colleagues in estimating the safe starting dose in humans. So there are different methods available, and 4 of the methods are highlighted here. The 2 being in this slide is NOAEL based method, which is a toxicological threshold based method, and MABEL, which is minimally anticipated biological effect level, where the safety is based on pharmacological threshold. And I'm sure that there is a talk coming up tomorrow, and I would say that that talk will elaborate more on this aspect. So just to briefly highlight that, you decide NOEL from your most sensitive species, convert that into a human equivalent dose by applying relative body surface area, and then divide this human equivalent dose by a factor of 10. And this factor is not a concrete factor. You can adjust this factor. Either you can increase or decrease based on the dose response relationship, which you might have seen in your early toxicological studies. And then you go for setting the safe starting dose in humans. Again, this is not a straightforward exercise because the requirements for oncology molecules completely differs, because for non-oncology indications, you often would do the study in healthy volunteers. But for oncology molecules, you do the study in patients, and that's why the dose which you select for non-oncology studies should not show any pharmacological effect. But when you go into the oncology indications, where you will be testing in patients, where you do not want to test a molecule in humans without showing some effect, and that's why it's very crucial for you to differentiate how you calculate the safe starting dose for a non-oncology and oncology indications. And also, it also varies for the monoclonal antibodies, other biologics too. And then the remaining 2 methods are pharmacologically active dose-based method and the PK or PKPD approach, where you see the emphasis is more on concentration and not on the dose. So finally, a little slide on the contribution expected from toxicology and regulatory dossier. And I will not go through the list. You can refer to this later. And I would like to finally summarize my talk by again reiterating the need of adopting innovative toxicological testing strategies in the routine toxicological testing methods available, with an aim to come out with a better translational outcome so that we avoid surprises when we go into the first-in-man trials. And some of the key aspects of toxicity testing where there are a lot of scopes lies in terms of improvement is listed down here, for example, from species selection to route of administration to volume of administration, for example, the formulation which you choose to increase the exposure, and the doses and dosage regimen, and also the TK is very important, the toxicokinetic part, where you want to show a proper dose response curve, And last but not the least, that you are always at your liberty based on a science-driven thinking to include some additional measurements or endpoints or biomarkers with a motive to reduce the risk during the first-in-man testing. With that, I would like to thank you all for your attention and open to answer any questions.
Thank you, Dr. Senthil Kumar. We've got a couple of questions over here. The first one is, as we have to establish two NOELs, one from novel rodent and the other from non-rodent, typically under IND enabling testing strategy, which NOAL would be appropriate to move for projecting the first-in-man dose for initiating the clinical trials?
Yeah, that's what I was trying to explain. I mean, that you have to consider the most sensitive species out of the two. And there are different ways to calculate from a rodent and non-rodent species, because, for example, from a rodent NOAL, you will convert that into human equivalent dose by using the appropriate body surface area conversion factor, for which I would suggest to refer to the FDA guidance on first-in-human testing available. And then you apply a factor of 10 to it. And then you also should see the dose response observed in your pharmacological, or the pharmacological as well as the toxicological testing. That if the dose response is very narrow, then you really need to apply the factor to a larger extent, not necessarily 10, but you can go for a higher value. And if a dose response is very, very broad, for example, from the effective concentration to the toxic concentration, you see a very wide margin, then you are at your liberty to narrow down the factor. So ultimately, it comes to the matter of most sensitive species. And you also should take into consideration whether the species which you select is pharmacologically relevant or not, because it's not always true that both the rodent and non-rodent will always be pharmacologically relevant. Either one species will be pharmacologically relevant. So there are so many aspects which need to be taken into consideration, by which you have to take the best dose out of it to go into the first-in-human.
I think that answers the point. The second question is, which bridging studies do you perform for switching clinical route from intravenous to inhalation? Maybe from
that's a good question. Yeah, first to start with the PK, I would say that you need to do a bridging PK by the new route of administration, for example, inhalation in this case, because already all the data were generated for the intravenous route of administration. So first you will do a bridging study, PK study for the inhalation, and compare the exposures there. Both, of course, for the inhalation, you will have to consider about the local effects as well. But when you compare the exposures, then you will have the from that's the starting point, I would say, to proceed further into the further toxicological testing.
Thank you. So one more question we can take up, I think, from here. 2020 saw a steady rise in silico trials. How relevant these trials are in future?
Yeah, that's what I wanted to come into the revolutionary aspect of toxicological testing, which is currently going on, both in the early safety assessment part and to some extent in the regulatory toxicology aspect as well. But these are all currently under trials, and I would say that a lot of improvement has been seen in the recent years. But when it comes to the matter of regulatory toxicity testing, it's still to see the light of the day. I would say that maybe in the years to come, we might see that the agencies might adopt or approve such in silico or in vitro trials as a replacement for some in vivo toxicological testing. But yeah, at the moment, as we are talking, we are still to see that happening. So currently, we still have to rely on animal testing. And for early safety assessment, you can rely to a larger extent on the in silico trials. For example, the QSAR, plenty of data is available. And you can, if put together properly, you can have a very good predictive toxicology package, which would really avoid some surprises when you go into the regulatory toxicity phasing and further into the first in humans.
True. These tools can only be used for prediction, not to make your conclusion. I think.
Yeah, exactly. Yeah, that's true. Yeah.
Thank you. Thank you very much, Dr. Senthil Kumar.
Thank you.
We go on to the next speaker. Dr. Nigel Room. He will be presenting a case study. Nigel Room is a pathologist and toxicologist with Sanofi or its previously incorporated companies. He is currently an independent expert in toxicology and toxicologic pathology, working with a wide variety of small and large pharmaceutical companies, non-governmental organizations, and consulting firms on scientific problem solving for toxicology and pathology issues, dossier preparation, review, and submissions, in addition to due diligences and interactions with regulatory agencies. He is also involved in teaching on several European and international programs. He is based in Paris, France. Dr. Nigel Room, you can initiate your presentation.
Hello?
Yes, we are there.
Can you hear me?
Hello, we are not able to hear you.
Can you hear me?
Yes, but I think there's some break in your voice sound.
Dr. Nigel, are you able to share your screen? Can you hear us?
Dr. Nigel, you are on a mute. I kindly request you to unmute yourself. Can you hear yours?
I think now we can see your screen. Yes, you can proceed. And then you can put it in presentation mode first. Hello, we can't hear you.
Can you hear me?
Yes, we can hear you. So now you can put this.
Sorry, there are too many. There are too many icons to do.
You can put your slide in the presentation mode now.
Oh,
Yeah.
hang That's what I'm trying to do. Can you see it?
No, we can see your screen, but not the presentation. Yes, now we can see the presentation. Can you put it in?
You got it?
Yeah, can you put it in the presentation?
Okay.
Mode,
Well, I'm trying to put this.
Dr. Nigel, in that case, do you want us to share the screen?
You got it?
From our end, yes.
I've put it in presentation mode. Can you see it?
Yes, that's
Hello?
fine.
Yes, we can see the slides, but it is not in presentation mode.
Well, on my screen, it's in presentation mode. I'm sorry, but I've got full
Manisha,
screen.
Can we help him by putting it the presentation which you have so that we can take him through?
Yes, I can do that. So you, Dr. Nigel, can you stop sharing? I will share my screen for you.
Okay. Is that okay?
I think you'll have to stop sharing the screen so that Manisha can carry your presentation.
Is that okay?
Yes.
Akila, I can't share the presentation. Maybe I'm not the co-host at this moment. Can you do that for me?
No worries. Can everyone see my screen, please?
Yes, we can see your screen.
Yeah.
So now we can put this into presentation mode.
Yeah. Is it okay now?
Yes.
Okay.
So Dr. Room, you can start your presentation now.
Thank you. I don't know why it wouldn't work. I'm very sorry about that. Okay.
No problem.
Myself, Dr. Nigel Room. I'm working as a consultant with DNDi for several years, and before toxicology pathologist. What I'm going to present here is a case study, second slide, on a review of the NOAEL and safety margin in the non-clinical studies performed on a product where there was an issue with an unexplained death in a monkey. So the background, there was a toxicology package prepared. There were two 28-day oral repeat dose IND enabling studies in the rat and non-human primate to establish the NOAEL. There were no toxicity or adverse findings considered in the rat up to the highest dose of 25 mg per kg, and the NOAEL corresponded to this dose. There was no toxicity or adverse effects in the NHP up to the highest dose tested of 15 milligrams per kg, but unfortunately there was an unexplained death with no clear cause seen in one of the animals at 15 milligrams per kilogram. So this compromised the fixing of a satisfactory NOAEL for this species. And as normal, obviously, the NOAEL would be fixed across the species to use the clinical doses to be tested in phase 1. So for the monkey, the NOAEL was reduced, and that reduced the overall safety margin for going into phase 1. So the problem was to try and resolve the issue of this unexplained death. If we look at the data for the two 28-day repeat dose toxicity studies at 6, 12.5 and 25 in the rat, there were no real adverse findings, no target organs really identified at the doses testing. The changes seem only minimal. With the exposure margins, you can see here, the exposure data you can see here on the right-hand side of the table. On the 28-day primate study, the doses were 2.5, 5, and 15. The exposure was less than in the rat study. And when the NOAEL was fixed at 5, due to the unexplained death at the high dose in this study, obviously the safety margin was considerably reduced. The first thing that was done to try and explain this unexplained death was to go into a non-GLP investigational study 28-day study. At 15 and 30, it was decided to go at the same high dose as the NHP study and to do a higher dose. And in this study, up to 30 milligrams, there were no effects and no unexplained deaths and no expected deaths. So in fact, we did not find the problem we had in the initial 28-day study. If you look at the safety margin, this is the next slide, the safety margins that were shown with the AUC values. Obviously, because we had the problem with the unexplained death in the monkey, the NOAEL was reduced to 5 milligrams per kilogram, and the phase 1 single ascending dose study based on stopping rules was at the exposure margin in this species. So that was an effect that would have blocked going higher in the clinical trial. And because it was intended to try and go higher to see the efficacy of the product, the idea was to go and see if we could find a reason why we had this unexplained death. If you go to the next study, next slide, if you look at the data we have here for the reevaluation of the sacrificed female on the 28-day GLP repeat dose toxicity study, there was the one explained mortality on day 22 of the 28-day study. The animal showed a quite marked body weight loss, low temperature. There was dehydration, inflammation, stress, gastrointestinal fluid loss. And the conclusion of the study was that the cause of the poor health of this female could not be explained, as no test item related histological findings were noted at necropsy. Obviously, this is always a problem for regulatory agencies, as they do not like unexplained deaths on repeat dose toxicology studies, especially if they are in non-rodent species, and particularly if they are in monkeys. And unfortunately, the NOAEL for females was fixed at 5 milligrams, and this was used as the AUC for the stopping rule in the single ascending dose study. This was in 2018. There was a great deal of discussion about the finding in this study, about how to address it. And it was decided to review the overall data from the dossier studies, and in 20 consultations, see if there could be a reason why we had this unexplained death on the study. And an additional external consultant suggested that there might be a reason to explain why it was a non-test item related observation. And it was requested that the pathologist on the study went back and reviewed the data overall on the study, and particularly in the animal that was sacrificed on day 22. Now, inflammatory changes were observed in the colon and cecum, and based upon the data which he reviewed, it was considered that these were consistent with spontaneous changes that had been described in the literature in both cynomolgus monkeys and rhesus monkeys. And in that case, the poor health seen was likely incidental rather than being treatment related. This has been described in the literature, as you can see, especially in the article of 2018, as an idiopathic colitis, which is considered as diarrhea and inflammatory changes in the GI tract, unrelated to test item toxicity, and can occur in both treated and both untreated non-human primates used in toxicology studies. It was decided in that case that the IB would be updated based upon this data to request a review to increase the NOAEL for the non-human primate from 5 to 15 milligrams per kilogram per day. If we look at the actual data that was present, and this animal at 15 milligrams was sacrificed moribund on day 22, there was no macroscopy, there were no treatment-related changes considered. They were considered at the time to be related to stress in adrenal glands, the thymus. The other changes were considered to be spontaneous and unrelated to treatment. Large intestine with acute inflammation, minimal, and slight effects in the kidney and sciatic nerve. The conclusion at the time was that the changes were related to poor health, and the cause of the sacrifice of an animal could not be determined. If we look at the changes that were actually seen in the colon and the cecum of the animal, there were effects, as you can see here, in the colon with inflammation at the higher portion of the glands within the colon, and were also seen within the cecum. There was also a hypertrophic, hyperplastic change within the colon and the cecum. If you compare to the control animal on the right, as you can see in the slides, compared to the treated animal on the left, there was an effect, an obvious effect on the mucosa of the gut. If we go to the next slide, the colon showed glandular dilatation, desquamation, accumulation of mixed inflammatory cells, and these were indicative of recent acute changes and may have been of a further longer duration, subacute changes. The inflammation was probably more acute, but the actual thickening of the mucosa was probably of a longer duration. So the animal had probably had a subchronic effect that was occurring in the gut, which would be consistent with what was described in the literature. And if you look at the literature, which is described idiopathic colitis in the rhesus macaque with dysbiosis, enterographia cells, altered T-cell cytokine expression. The minimal acute inflammation was originally recorded in the colon and cecum, unrelated to treatment. The relation to poor health and premature sacrifice was reconsidered, and this paper was used as justification for why the effect was not considered to be clearly not treatment related, and effect of the treatment could be excluded.
Dr. Room, you have got 5 minutes to complete your presentation.
Yeah, we know that chronic diarrhea is a major cause of morbidity in rhesus macaques. Can be infectious cause with enterocolitis, and up to 25% of rhesus macaques can have an apparently non-infectious diarrhea syndrome, which is either referred to as idiopathic chronic diarrhea, and the pathogenesis is still obscure. It can be persistent, it can recur with clinical signs, and it can lead to repeated hospitalization, in some cases requiring euthanasia. It has been described, as we have here, in cynomolgus monkeys used in preclinical development. Based upon the review of the data, the pathological lesions that were seen, it was considered that the effect was probably not treatment related. So the final conclusion, following review of the literature and the rereading of the slides for the 28-day repeat dose toxicity study, was that the death of the animal was not linked to treatment with the compound, and thus the NOAEL from the study could be raised from 5 to 15 milligrams per kilogram per day, and thus you can see that there was the possibility to increase the dose limit that was fixed in the phase 1 study from the initial value that was given based upon the 5 milligram per kilogram dose up to 15 milligram per kilogram dose. So this indicates that it is always useful to go back and try and look and see why, especially when you have an unexplained death, if you can find a reason why this animal would have died. It occurs fairly often in toxicology studies that you have unexplained deaths. I have seen toxicology studies where dogs or monkeys appear to just give up in toxicology studies. It's not necessarily linked to the toxicity of the product, but they can have concomitant infectious effects, they can have concomitant other effects. And unfortunately, you have no pathological lesions, you have no reason why the animals should be sacrificed or died, especially in the non-rodent species. And it's always very important to go back in and look and see if you can explain why you have these unexplained deaths. Thank you very much. If you have any questions, I'm willing to take them.
Thank you, Dr. Room. I think we can take up a couple of questions here. The first question is, does it make sense to go back to the rat and look at this colitis between treatment and controls to help substantiate, because it's becoming too expensive or too time consuming?
No. Well, there was nothing. This is not a lesion that's described in the rat. This is purely a non-human primate lesion, and there was no toxicity in the rat, and there were no deaths in the rat, unexplained deaths. So from that point of view, as you can see in this final slide, the NOAEL was considerably higher in the rat because there was no toxicity, absolutely none, no unexplained deaths, no toxicity, no organ toxicity at all. So the problem was the NHP, was the non-human primate, which was causing the issue. So there was no need to go back in and look at the rat. And this is not a lesion that's described in the rat as a spontaneous background change.
One more quick question, just inquisitive, to know, would you have applied the same approach if at all there was such death in rats? Because there, if you account for the sample size, the percentage would have been very low vis-a-vis the monkey studies.
I mean, if you had the same effect in the rat, it could be a treatment-related effect, obviously, which wasn't the case here. There is no indication as to why this compound would cause a gastrointestinal toxicity. So obviously, if you had a similar effect in the rat, you would go back in and check, and you would look at your receptor binding, you would look at the activity of the product. Would you have an indication that the compound had a gastrointestinal toxicity? And in that case, you would reassess the overall NOAEL in both species and see which was the less sensitive, and would fix your starting dose and your maximum dose that you would give in your single ascending dose study in humans based upon these data. But that was not the case here. Obviously, if you had dose-related toxicity in both species in the gut, you would go back in and look, but that was not the case here.
Thank you, Dr. Room, for the case study. I think we can go to the next case study by Dr. Stephanie Ballard. She works with the non-clinical development senior as a non-clinical senior manager with DNDi since 2019. She is responsible for all non-clinical matters through the portfolio as soon as the compound is entering into the preclinical phase. Before that role, she was a part of the DNDi discovery team and has contributing to lead optimizations and preclinical projects. She previously served as a scientist in various biotech companies in the fields of antibacterial drug discovery and development of treatment for onco-urology diseases. Ms. Ballard has obtained a master's degree in molecular and cellular biology from the University of leucine. So, Dr. Bernard, you can initiate your presentation.
Thank you very much. I hope that my slides are visible and that I'm audible.
Yes.
Yes.
I think your
Thank you very
slides
much.
well audible
So
also.
good evening or good morning, good afternoon, where you stand in the world. So really pleased to present this case study. And actually, just to give you a little introduction or background on that case. Actually, it's an example that can happen actually when you have to deal with a potential and known target organ for a particular chemical class you are working on. And actually, it can have an impact on your development. And this impact can stand there from lead optimization to first in man and even probably beyond, because any finding, any observation will stay in your dossier. And if I can resume actually with one sentence this case, I would use that one. When you look for something, you will likely find it. And you will understand why I'm saying that a little bit later. So just to set up the scene, here we are talking about a chemical series that we will name lambda, that is known actually for a risk for testicular toxicity. And it has been seen in several molecules from that chemical class in different species, not always in all species that were tested, possibly in man. It was seen sometime with reversibility, sometime without reversibility. At different level of exposure following various treatment duration. And from that chemical class, actually, two compounds just pop up, lambda 42, lambda 43, that were identified as very promising candidates for a particular disease area where actually testicular toxicity will not be something acceptable for the patient population, and also as a very risk that is difficult to monitor in the clinic. And let's see what happened to these two molecules from the lead optimization till the first in man. So you probably know that the three big steps when you're at the early stage of development are the lead optimization, followed by preclinical development, and obviously first in man. And for this different step, you may have different goals when you are working on a target organ. So when you're at the end of the lead optimization, when you have to optimize your lead, maybe the first goal here would be to try to discard any risk. And here in our particular case, to discard the risk for testicular toxicity. And you want to do that because you don't want to invest in the preclinical development that is very costly if you have a risk. Then actually in the preclinical development, if the risk is still there, you will continue to monitor it, to characterize it. It's just qualitatively, but also to see if it's reversible and understand about the safety margin. Finally, when you are reaching the clinical trial, the first goal that you have is absolutely not to put at risk your healthy volunteers, to continue to monitor actually the risk and do this, what we say, this recurring assessment of benefit risk. That is a really an exercise that you do again and again. And for all this step, you have to establish a plan to reach that goal, from which you will have some outcome. And after each step, the outcome could actually change and refine the plan for the next step. And I will illustrate that with this example on testicular toxicity, starting here at the lead optimization phase, where, as I say, we wanted to discard any risk for testicular toxicity. So the plan that we have established was to start with a 2-week study in the rat. And actually, we have tested the two compounds in parallel using the same formulation. And we have tailored the study, making sure that if we had to face any testicular toxicity, we would see it. So for that purpose, first, we have guaranteed the quality of the study. So we work under GLP, something that usually we are not doing necessarily at this stage. We have also planned for a long period of recovery, so 4 weeks actually. And this period of recovery was actually assessed for all the different doses tested. This was actually the aim of that was to cover the spermatogenesis cycle in the rat in case of findings. Obviously, we have used sexually mature animals. We had 5 female per group. However, we have increased the number of male just to have a more power in that study. So we had 10 males per group. And finally, obviously, we have done some histology on a specific organ, reproductive organs in that case. And we have asked an expert in testicular toxicity to peer review the pathology. What was the outcome? Actually, for the first compound, lambda 42, we have seen clear testicular toxicity. We had minimal and reversible finding at the mid dose and a very obvious finding at the highest dose. So it was clear that testicular toxicity was there, nothing at the low dose still. But lambda 43, much better because no observation at all doses for any kind of testicular toxicity. So outcome of that obviously was to stop any development for this first compound. For the second one, however, this was clean and we had the green line to continue to the next phase, the preclinical development. So let's see what happened to the preclinical development of lambda 43. So as I say, goal here is really to continue to monitor for the risk of toxicity if present, because if not seen in the rats, the non-rodent species were not yet tested, so better to continue to look at it. So the plan was first to have 2 studies in the rat, the MTD first, where we have tested higher dose level than was previously tested in the exploratory tox. And we have done the pivotal study, again, 4-week GLP with sexually mature animal, and again, the 4-week recovery period. In parallel, the non-rodent species was actually the non-human primate in that case, where we have conducted the maximal tolerated dose followed by the 7-day dose range finding. And as well for the pivotal study, 4 weeks, we have used sexually mature animals. And in that study, again, we have used a long period of recovery in case of finding that was 7 weeks, again, to cover the spermatogenesis cycle in that species. Outcome of what we have from the rat study, absolutely no observation of testicular toxicity again, even if highest dose was tested, longer duration. And finally, the highest dose appeared to be the NOL. For the monkeys, it was a little bit different. First study, the non-GLP-1, actually the 2 out of 2 male were affected by actually the fact that we have seen vacuoles in the testes. In the GLP study, and this is illustrated in the little picture there, we had some finding as well. In 2 out of 3 animals in the mid-dose group, we have seen bilateral tubular atrophy, even hypoplasia. It was exactly the same at the high dose group with the same severity and the same number of animals. And finally, in the recovery group for the high dose, we have seen still some findings, less severe, but still there in 1 out of 2 animals with this bilateral tubular atrophy. So the conclusion we had at that time that first for that specific study in the monkey, still the lowest dose was the NOAL, that if you look at the full picture for the monkey, we have a kind of clear treatment duration dependency, a trend for recovery, but not full recovery. And it was difficult to find a relationship between exposure and an event. And at the end, we found that we had a manageable safety margin still higher than 10. And the outcome of that was, okay, let's continue to phase 1 in man, but there is a clear flag there. So first in man. Here again, you remember that I say that the main goal is to take absolutely no risk for the healthy volunteers, but in parallel to continue to monitor, to better characterize the risk you have observed. And the plan for the phase 1 was actually the single ascending dose was performed in the healthy male subject mainly, still with 1 cohort of women, of course in non-child bearing potential. And we found absolutely that we are taking absolutely no risk at all for the healthy volunteers regarding testicular toxicity, because this was obviously a single administration dose and there is no risk after 1 single administration for testicular toxicity. And following that, also we haven't monitored at all. This is something really difficult to do in the clinic for testicular toxicity. And actually, the study has been completed and was absolutely successful. In parallel, actually, and this concerned the non-clinical aspect. Actually, a bit by chance, an opinion was gathered from an expert, an opinion that actually was at that time gathering and reviewing data from control animal regarding testicular toxicity. And when he looked at our data coming from the monkey, he said, well, maybe what you have here could be background lesion only. And I would recommend that you reassess that. So this is something what we have done by setting up a pathology working group. A pathology working group being when you convene a few pathology experts, independent experts that don't know your project, who don't know your data, and look at your slide, probably in blind mode, and just have their own opinion, then go together and find a consensus. And actually the
Stephanie,
outcome...
you
Yes?
have 5 minutes to complete your presentation.
That's perfect. Thank you. And actually, the outcome of this pathology was at the end, in the first study, so the non-GLP, the vacuolation we observed was only an artifact of the fixation. And in 20D, what we observed actually was not at all bilateral, or if bilateral, only peribilateral, so something that you will observe in not fully mature animals, or only unilateral, and no observation at all in the recovery group. So this means that here, the conclusion of that changed dramatically. And it was actually that the testes were no more a target organ for this compound 43. So from that, from the successful results of the phase 1, this new assessment of non-clinical data, actually we had now a clear and clean path toward phase 1 MAD. So just to conclude on this case study and maybe a bit of lesson learned, the first thing I would say is that pathology and toxicology science are ever evolving. It can be that new research, discoveries, or reviews can change and change very significantly. You have seen that here, the conclusion of your toxicology study. Second thing, pathology remains something that is inevitable human and a subjective bias. So it's always very, very valuable to have several opinions. This can be done by peer review, for instance. Maybe new with the new technique that is coming actually with artificial intelligence, maybe this will improve, but let's see. And actually also when you have a target organ that you are really seeing here, and popping in, what you have really to do, actually, it's not only to focus on doing everything for make it shine, something that you have to do really to demonstrate that if it has to happen, he has to be showed. And this is what we have start from the beginning, if you remember about this ExploTox that we have tailored in that direction. But second things is that when you have findings that pop up, don't say that, okay, it was accepted, it's normal, but I would rather double check and challenge also these findings. Voilà, thank you very much. I'm happy to take any question if you have some.
Thank you, Stephanie, for your very intriguing presentation on this case study. There may be a couple of questions I would like to go through. One is, what are the parameters that were assessed in human to conclude drug is not testicular toxicant?
So as I said, this is something that we have not monitored actually at all, because this is very, very difficult to do it in a clinical settings. And also it has a lot because of variability between subjects, etc., etc. The number of subjects that you will need to do that with accuracy and robustness, it's really difficult. So this is something that we have not assessed at all in the clinic. And actually, it was not necessary from the MAD study, because testes were no more a target organ at that time.
So from the case study, it can be concluded that the toxicity that is seen is basically specific to that particular species. Am I right?
It can be for that molecule, but it appeared to be not a target for any species in that specific case. For other molecule, for the same class, we have seen that sometimes it's a target organ for a species, not for another one. Yes, correct. It's really very random, I would say.
One more question. I don't know how relevant it is for the presentation. Are there particular learnings you can share for pediatric requirements?
For pediatric requirements, so for sure, we will have to be careful for pediatric population in the future for the development. And especially what we are looking now, because for that the preclinical package, as you may have seen, we have make sure that we have involved sexually mature animal in our trial. So this means that animal of lower age were not covered. So now we are doing an assessment if a juvenile toxicity, for instance, will be necessary for the future development of the molecule.
I think there are no further questions. So thank you, Stephanie, for your
Thank you, my
wonderful
pleasure.
presentation of the case study. So as we come to the end of this session, on behalf of the organizers, I take this opportunity to extend my gratitude to all the speakers for a very lucid, intriguing, and thought-provocative talks. I also thank the organizer, DNDi, and its associates for setting up this informative platform, and the team who have worked and coordinated relentlessly for a smooth flow of activities. Last but not the least, I would like to express my appreciation to the very interactive audiences who, by the presence, have added value to the webinar. The audience is requested to fill up the feedback form posted in the public chat. The form link will be live till another 15 minutes after the workshop and will close. So this is a request. Thank you all. And with this, we sign off for the session. So Manisha, any closing remarks from your side?
No, sir. Thank you so much. On behalf of DNDi, ISCR, and BRBC, I thank you and all the participants and our panelists for taking out time to attend this workshop. Thank you so much. Hope to see you all tomorrow, which is the concluding day of this 5-day series. Thank you so much. See you tomorrow. Bye-bye.
Request all to kindly fill up the feedback form. The link has been put up in the chat box.